All right, when we look at what we consider a synapse, by definition, what a synapse is a neuron, a meeting place, if you will, between a neuron and another neuron, or a neuron and another, if you will, effector cell. In chapter 10 in AMP1, we talked about how that synapse was called a neuromuscular junction. It was a meeting place between a neuron and that effector cell happened to be a skeletal muscle. All right, so that's what we called a neuromuscular junction, but basically what it was a synapse. All right, so again, by definition, a synapse is a meeting place, a place of contact between one neuron and another neuron, or it could be an effector cell. There are different types of synapses. There's a synapse called an electrical synapse, where there's just actually going to be a meeting place between a neuron and another neuron, but there are gap junctions between those cells. So whatever happened in this cell, which is, let's say, an action potential propagated down the cell, it will go right through that gap junction to the next cell. All right, and so those are really fastly transmitted signals, obviously, all right, because it won't have to go through the next type of synapse. It won't have to go through all of the things that a chemical synapse has to go through. And that's what we're going to focus our attention on. Pretty much most of your synapses are chemical synapses. And so it starts where you have an action potential go down, if you will, one neuron, the actual axon of it. Once that action potential gets down to what you consider the axon terminals, okay, then that particular action potential is going to actually stimulate voltage-gated calcium channels down here, okay? That's going to cause calcium to rush into those neurons and cause what we considered in lecture these vesicles that contain neurotransmitters to exocytose those neurotransmitters. And so that's how calcium is very, very important in this process of what we call a synapse. So based on those voltage-gated channels, okay, opening, as a result of that action potential coming down there at that axon terminal, or axon in bulb, that's the most terminal portion of an actual neuron, axon in bulb. You're going to have calcium now influence vesicles that contain neurotransmitters to exocytose that material. And now what you're going to have coming out right here is a whole bunch of neurotransmitters that end up going outside of this neuron now into this space called the synaptic cleft, okay, which is a major part of what we consider a synapse. Synaptic cleft is the actual fluid, the space that has this fluid which is called interstitial fluid, right? Remember from AMP1? The actual fluid between cells, right? That extracellular fluid is called interstitial fluid. So what you're going to have is this actual neurotransmitter go across, okay, across that actual synaptic cleft from one neuron to the next, all right? And that traveling space there is very critical as it relates to doing what? Transmitting that signal that was on this neuron to the next effector cell or neuron in this particular case, all right? All right, so it's very interesting, and that's what's incredibly interesting for individuals in medicine, because it's at this location, that synaptic cleft, where we can actually change certain things. We can't change anything that happens to action potential on this neuron, nor this neuron here. But we can actually change things in this synaptic cleft area, where you're going to have your neurotransmitter traveling across. Okay, so that's a very special place there. What's going to happen next is your neurotransmitter makes it all the way across, and then binds to his receptor on the next actual effector cell, or in this particular case, the neuron, at where they are the dendrite or the cell body. All right. Once he binds to this receptor, okay, it depends. It depends on what that receptor is. If that receptor happens to be which we'll get into later, whether it is an ionotropic receptor or a metabotropic receptor. But in that case, if it is a channel, if it's associated with some sort of channel opening, let's say a sodium channel, then it may cause an actual depolarization right here. All right, depending on how many sodium channels open, that lets you know what the inside of this membrane potential is going to be. If it's a whole bunch of them, and it's a whole bunch of sodium channels that open up, then you may actually get an actual depolarization occur that goes past a certain threshold of what, -55, which we've talked about before. And if those voltage-gated channels here at the axon of this next neuron detect a -55 voltage, okay, millivolts, then you'll actually get a what? An action potential occur here. All right, and so that's what's very special. But what we talked about before was the fact that at dendrite and cell body portion of a neuron, you have what you call a graded potential before you get what you call an action potential at the axon level. All right, but yes, at this synapse, depending on how many actual neurotransmitters influence receptors here in channels, depends on what your graded potential is going to be. All right. All right, and so, but that's what's happening at a synapse. So let's describe what happened again. We had an electrical signal called an action potential travel down an axon of the first neuron. And then you're going to actually have that energy, that's electrical energy, be, if you will, change from electrical energy to chemical energy in the form of a neurotransmitter. And then you're going to actually have that neurotransmitter now cross that synaptic cleft and now interact and bind with its particular receptor, cause certain channels to open. And now you're going to have what? That chemical energy turned back into, if you will, electrical energy. All right? All of that is as a result of what we call a chemical synapse. And that's conveying signals from one neuron to the next neuron, or from one neuron to another what? Effector cell. Okay, so that's what we call a chemical synapse.